Hello everyone and welcome to the NPTEL course on pharmacognosy and phytochemistry. In the previous session, we have learned what are glycosides, the classification of glycosides and few drugs containing glycosides, that is aloe and senna. Those were the laxative ones. In this particular session, we will dwell into more potent glycoside derivatives, and those are the ones which act on our heart. So, these are what are called as cardiac glycosides. So, welcome to a session on cardiac glycosides. So, cardiac glycosides, if you see, are sterol glycosides. So, by nature they are steroidal moieties and they have a good ability to act on human heart. As they are glycosides, we understand that the steroids have been attached to sugar molecules which make them more absorbable and affect its ADME, that is the pharmacokinetic parameters. The pharmacodynamic parameters are attributed to the steroidal moiety. In the cardiac glycosides, we can divide these compounds into two categories, cardenolides and bufordinolides. The difference is their carbon count. So, if you see their numbering, the basic steroidal moiety, the numbering starts from ring A. So, steroidal moieties have a 4-ring system. A, B, C are 6-membered ring system, whereas ring D is a 5-membered ring system. So, numbering starts from ring A and goes in this manner. So, if you can see, it's a typical what we call it as anti-clockwise manner. So, you have your carbon number 1, 2, 3, 4, 5. Now, naturally when you have sterols, many of them have a hydroxy group present at the third position. So, 1, 2, 3, 4, 5, then 6, 7, 8, 9, 10. So, your ring A and B is a 10 carbon ring system with shared carbons, that is carbon number 5 and carbon number 10. Now, when you go to ring C, the numbering happens in a slightly different manner. As you can see, the numbering happens in this manner. So, you will see then after 10 is 11, 12, 13, 14, 15, 16, 17. Okay. So, now if you see between your ring B and C, the shared carbons are at the carbon 8 and 9, and between C and D, the shared carbons are carbon number 13 and 14. From cardiotonic activity, carbon 12, 14, 16 and 17 are very vital. We will see them as we elaborate more into cardenolides and bufordinolides. But just to give you an overview, cardenolides generally contain what is called as gamma lactone system, whereas bufordinolides contain what is called as a delta lactone system. So, here you will see them very nicely, the difference between the two. So, this are steroidal moieties. If you remember, you will have your carbon number 1, 2, 3, which I said was hydroxy, 4, 5, 6, 7, 8, 9, 10, and then onwards you can recollect 11, 12, 13, 14, 15, 16 and 17. Okay. Now, here the sugar is attached in a beta position and so is the lactone moiety. But in cardenolides, you will see a 5-membered lactone moiety, and in case of bufordinolides, you will see a 6-membered lactone moiety. One more thing, as you can see, a difference between is here the number of double bond is just 1, whereas in this case you have 2 double bond structures. In terms of their occurrence, cardenolides principally are found in plant species, whereas bufordinolides have been found in plants as well as certain species of frogs or toads. So, they are also found in the animal kingdom as well. Cardenolides, you can say, example of cardenolides containing plants include your digitalis, they include your Thievesia, they include your Strophanthus, whereas bufadinolide is present in your squill and buffa species of frog. When it comes to the glycone moiety or the sugar moiety, like I said, they have been attached to the 3-hydroxy linkages of the steroidal moiety, chiefly by a beta linkage. So, carbon number 3, you take a beta linkage and attach a sugar. Now, predominantly these sugars include your deoxy sugars. One of a good example of a deoxy sugar is a digitoxose, which is present in your digitalis. So, if you can see this particular carbon, okay, it's a deoxycarbon out here, whereas this is your glucose. Apart from it, some of them are also found to contain rhamnose. So, you have your deoxy sugars, rhamnose, you have your fucose, digitallose, tivetose, 2,6-deoxy sugars. This is 1, 2, but you might also have your 2,6-deoxy sugars such as digitoxose. Okay, you can see it clearly. Okay, and then cymerose and oleandrose are some of the examples of deoxy sugars. Now, glucose, when it is attached to this type of glycosides, it's mostly the terminal or the outermost sugar. Now, if you see glycones, I am just giving you few examples of glycones which are very potent as cardioactive drugs. And if you see in terms of substitution, let's come back here. Here you remember 1, 2, 3 is a hydroxy, 4, 5, 6, 7, 8, 9, 10. You are done with this. Then you have 11, 12, 13, 14, 15, 16, 17. I am just repeating it so that you all can visualize it more clearly. So, in case when you have a hydroxy at 14 position, now apart from 17 you have your 18, 19 as well. So, total 19 carbons in the steroidal moiety, but whenever a hydroxy is present at 14 group, we call it as digitoxygenin. Whenever the hydroxy is present at 14 and 16th carbon, you call it as gitoxygenin, and whenever it is present at 12 and 14, you call it as digoxygenin. Apart from that, you might also come across G-taloxane. Now, in G-taloxane, what happens is you get a esterification. So, this hydroxy is actually converted into a aldehyde. We call it as G-tal. That comes from gitoxygenin, the aldehyde of gitoxygenin. So, G-taloxygenin. Okay. So, these are few common glycones which occur in your cardiac active glycosides, especially in the digitalis species. So, let us see what is digitalis. So, digitalis is a beautiful Himalayan plant which grows in Europe or mostly the temperate climates. Depending upon the species, predominantly 2 species of digitalis are used, Digitalis purpurea and Digitalis lanata. So, Digitalis purpurea, let us talk about it. It is also referred to as a purple foxglove for its beautiful purple color flowers which represents or which kind of mimics your lady's glove, and it is belonging to the family Scrophulariaceae. It's commonly found and grown also as an ornamental plant in England, Germany, France, North America, in places India, Japan, Kurdistan, Mexico, Nepal, Spain and Turkey. Now, this plant, the leaves of it are very toxic. So, though it's ornamental, it's often handled with great care. Now, in terms of cultivation, what is done is in this plant the seeds are sown, and you will see in the first outgrowth it will bear what is called as a rosette leaves. Those are not very good or rich in glycosides. Subsequently, it will give you a nice ovate kind of leaves, and those are the ones which are preferred for its high cardenolide content. So, it contains somewhere between 0.1 to 0.4% of cardenolides, in terms of purpurea glycosides. So purpurea glycosides are nothing but your cardenolides plus sugars. Now let's break down this molecule one by one. So to see this, if you remember, whenever you have, now I am being a little quick, so you have this 10 carbons, 11, 12, 13, 14. So 14 hydroxy is, if you remember, it was digitoxygenin. Okay, so if you have your digitoxygenin attached to 1, 2, 3 digitoxo sugar, so I am just putting here plus 3 into digitoxos plus 1 molecule of glucose. This is what is your purpurea glycoside A. So purpurea glycoside A has a glycone digitoxygenin and a glycone moiety consisting of 3 molecules of digitoxos and a single molecule of glucose. This occurs primarily in the plants and are referred to as primary glycosides. Now what happens is there is also an enzyme present in this plant called as digipurpurase. So whenever the drug is broken, ruptured or due to some mechanical effects the leaves is bruised, in that case what is going to happen is the enzyme digipurpurase comes in contact with your purpurea glycoside and starts cleaving the sugar. So in the process you get this glucose being removed. So now you are left with 3 molecules of digitoxose sugar plus digitoxygenin, and this together is called as digitoxin. So digitoxin is a glycone plus 3 digitoxose when you remove a glucose molecule. Now this is referred to as secondary glycoside. So once the enzyme removes the glucose, you get this as a secondary glycosides. Now similar way your purpurea glycoside B will give you gitoxin and purpurea glycoside C will give you gitaloxin. Apart from that, your plant also contains what are called as steroidal saponins, flavonoids such as apigenin, luteolin and some cinnamic acid derivatives such as your caffeic and chlorogenic acid. So you can see here clearly purpurea glycoside A, remove the glucose, you get your digitoxin, remove this. Now how do you remove this? This is generally done by what is called as acid hydrolysis. Acid hydrolysis will give you glycone, but the problem is if you get your glycone, the glycone is going to be inactive because it doesn't have the required ADME effects. Now this care has to be taken during the harvesting stage. It is said that if you keep the leaves, so these leaves are generally harvested when the plant is in bloom or at least 3/4 of the inflorescence is open. So that time you collect the leaves having high cardenolide content. Now these leaves are to be dried immediately. The reason is if you don't dry it immediately, the digipurpurase remains active. And if the digipurpurase remain active, like I said, it starts cleaving the sugars, being the glucose being first. So enzymatic hydrolysis predominantly takes care of this, and to this point it is okay. But if further it deteriorates and it removes your digitoxose moiety as well, in that case you get a genin. And the genin has lot of issues in terms of its bioavailability and absorptivity. So it doesn't show the desired pharmacological effect, and as a result these leaves are not thought to be of good quality. So ideally the moisture content during the drying stage is brought to below 5% and then stored to prevent it from enzymatic hydrolysis. Similarly your purpurea glycoside B will give you gitoxin, which in presence of acid will get hydrolyzed to getoxygenin. Purpurea glycoside C will give you getalin. You remember the aldehyde substitution, and this will give rise to, on acid hydrolysis, getaloxygenin. So there are numerous tests. I have stated a few, but let's discuss them. So one of the tests which can be used for detection is your budget test. Budget test takes place, you can do it on leaf. So you just take a very thick section of leaf, and to that leaf you can add sodium picrate directly. When you add sodium picrate directly, it gives you a yellow to orange color. Alternatively, if you don't have sodium picrate, what you can do is initially add a drop of picric acid, and then to that you add an alkali like a sodium hydroxide. Eventually your picric acid will form a sodium picrate salt, and you will get the same thing. So it is said that the picric acid gives you a strong coloration, whereas a presence of base like a sodium hydroxide or sodium salt of picric acid in this case intensifies the color to a orange shade. The next test is a Legal's test. So in this case what you can do is you can take the pure cardiac glycosides, or you can dissolve or extract the drug in pyridine. Now this is treated little bit with sodium nitroprusside solution and made alkaline in a similar manner, but this time you will get a pink to red color. Now you can also do what is called as a test for digitoxose sugar, that is the test for deoxy sugar. In this case you can take the isolated compound, or you can take the plant extract. You can do an hydroalcoholic extraction, filter it, and then you add a good amount of glacial acetic acid. Now to that just add a drop of ferric chloride solution, and from the sides gradually add concentrated sulfuric acid. Now we know that concentrated sulfuric acid, owing to its density, will form a layer at the lower side of the test tube. So gently see the color change. So initially the interface between your ferric chloride glacial acetic acid layer and your sulfuric acid layer will be reddish brown in color, and slowly the ferric chloride and glacial acetic acid layer acquires a bluish coloration. So if this happens, it's a good chance that it contains a deoxy sugar. Apart from that, you also have Cade's test. Cade's test also helps you distinguish between cardenolides and bufadienolides. So what you do with that is you extract your plant, or you dissolve your compound and treat it with small quantity of Cade's reagent, which is actually a mixture. It is, you have to take equal quantities of 2% solution of 3,5-dinitrobenzoic acid in methanol and about 7.5% aqueous KOH. So when you take this in an equal ratio, you should get a bluish violet coloration, which gradually takes time to fade, like 1 to 2 hours. If no color change is seen, there is a high chance that it's a bufadienolide and not a cardenolide. You also have other tests such as your Raymond's test. Now, where do you apply this cardioglycosides? Cardiactive glycosides are known to act on a specific system called as sodium potassium ATPase pump. They inhibit the sodium potassium ATPase pump, and as a result what will happen is you will see a increase in intracellular amount of sodium as well as calcium, and this calcium in the cardiomyocytes or the heart cells as you call it will increase the contractility. This contractility is referred to as a good inotropic effect. It also exhibits a good thing on a good effect on a tone. It's called as tonotropic effect. So good inotropic, it maintains the muscle tone, and this contractility helps us in a way that, and since it's a better contraction, more cardiac emptying will happen. Now, so imagine a case in a congestive cardiac failure where your heart is unable to pump in. This gives you more energy to pump it, more vigor, and as a result the heart performs better. Hence this class of compounds are also referred to as cardiotonic agents. Apart from that, they also show a mild diuretic activity, which is also said to be as a result of its cardiotonic effects. Now we go on to another plant which also contains cardinoloids, and that is your Digitalis lanata. Unlike purpurea, which had a purple color flowers, as you can visibly see, this one has a little brown to yellow color inflorescence. So it is obtained from the dried leaves of Digitalis lanata, which belongs to the Scrophulariaceae family, and it's again found almost similarly in Europe, England, California and India, but you can see there's a notable difference between the leaves and the flowers of this plant. There's also a good difference between the chemical constituents. In this case, apart, you do not find your purpurea glycoside, but you find compounds called as lannatosides. Now, what is the difference between purpureoside and lannatoside? So, if you see this carefully, this is a lannatoside C. Okay. Now, the glycone here is degoxygenin. Degoxygenin is absent in purpurea. So, this glycone doesn't happen or doesn't occur in your Digitalis purpurea. Now, apart from that, you have your 3 sugars which are digitoxose, and 1 sugar which is a glucose. But at the third position, so this is like 1, 2nd and 3rd. At the third position of third digitoxose sugar, you will see an acetyl substitution. So that's the major difference between your purpurea glycoside and lannatoside. So in lannatoside, the third position of the third digitoxose sugar is acetylated. The glycones remain more or less same, but there is an additional glycone which is found in lannata and not in purpurea, and that is your degoxygenin. So you will have primary glycosides, lannatoside A, B, C and so on. You will have your secondary glycosides. Secondary glycosides in this case will give you acetyl because this is not gone, but this later on this acetyl can also be hydrolyzed. So after the acetyl is hydrolyzed, you will get your digitoxin, jitoxin and digoxin. Yes, hydrolysis of this acetyl group is also possible. Now then, in the end, when complete hydrolysis is done, you get the corresponding genin. So you will get digitoxygenin, jitoxygenin, degoxygenin and so on. Okay. Apart from that, it has other moieties also, like your diginatigenin, which is coming from your lannatoside C, and gitaloxygenin. So apart from cardiac glycosides, you have your steroidal saponin derivatives, flavonoids, apigenin, luteolin. Again, you have your cinnamic acid derivatives and some anthraquinones which are also present in this plant. Now, applications are very similar. So, it has a positive inotropic, tonotropic effect. It is a cardiotonic, so acts by inhibiting your sodium potassium ATPase pump, but the presence of digoxin has some differences. So you will see in market you have digitoxin and digoxin. So digitoxin and digoxin both are marketed and they have a slightly different application. As compared to digitoxin, your digoxin has a rapid onset. So the onset is as far as 15 to 30 minutes and it peaks also quickly. Within 3 to 6 hours it reaches the optimum concentration. So whenever a rapid onset is desired, in that case your digoxin is preferred over digitoxin. Digitoxin takes a little more time. Now one thing about this is your digitoxin is cleared mostly through hepatic clearance, whereas your digoxin is cleared exclusively by kidneys. So people, those who have renal failure, kidney failure, in that case you have to be very careful while handling digoxin as it may not get eliminated quickly because of the kidney malfunction. Again, a difference lies between the doses. If you see your digoxin is effective at a very low dose. You can say like 0.5 to 2 nanogram per ml of body fluids, whereas if you say digitoxin, it has a little wider nice margin, that is 10 to 25 nanogram per ml. So even if you see toxicity of digoxin occurs very fast, and hence this given very carefully under the supervision of a practitioner or a basically under hospitalization. Whereas digitoxin in some cases is prescribed even as oral tiny tablets for long term dosage whenever required, because it has good sustainability and a very wide margin of safety as compared to your digoxin. Now, if you go to see your bufadienolides, a good example of a bufadienolide is squill. Squill is onion-like sliced bulbs which is obtained from Erginia species, Erginia maritima, and if you go to see Indian squill, it's Erginia indica. Okay, so from that what they do is the outer sheathing scales have been removed. This is onion-like, so belongs to the family Liliaceae. It is cultivated in Mediterranean countries and West Asia. The white squill, which is the medicinal squill, is cultivated in Spain, Portugal, Sardinia, Malta, Cyprus, Greece, whereas red squill, if you go to see, is more native to Algeria and Morocco. Now, red squill is considered to be a little toxic and not preferred pharmaceutically. In terms of chemical composition, as you can see, it's a bufadienolide. It has got a 6-membered lactone with a 2 double bond. In terms of sugars, as you can see here, this is one molecule of glucose and one molecule of rhamnose, which is attached to the 3-hydroxy moiety of the sugar. Now, apart from that, it also contains an enzyme, like how digitalis had digipurpurase. This one has skeletonase, which causes the hydrolysis. Now, once it causes the hydrolysis, the glucose is lost, and what you get is what is called as proskylarin. Okay, and then you have another acid hydrolysis, so you get your genin being formed. So it has sclerenin, so it has erglycone, sclerenin, glucosclerenin, and especially prosclerenin. The red squill, which is the toxic variant, contains the principal glycoside, which is scleroside and glucoscleroside. They are erglycone part, which is referred to as sclerocidin. Now, this is the one which is sometimes also referred, or certain species of red squill are also been used to poison the rat. So, it was very famous as a rat poison initially, but it is no longer used for it. Apart from that, it also has other flavonoids and some derivatives called as xanthoscleide. Applications, if you see sclerin or proscleridin A, especially from white squill, which is the pharmaceutical one, which gets easily absorbed from the GI tract and hence given as a oral dose as a therapeutic in number of cardiovascular conditions such as this includes aorta insufficiency, angina pectoris, edema, which is also associated with nephritis. And a counter effect of all of this is, it is seen that when given in small doses, it can take out a cough and is used as expectorant, but if you just administer it in a large doses instead of cough, there is a good chance that it will cause a vomesis. So, this is with your cardenolides and bufadienolides, the chiefly cardiac acting drugs, and here's if you wish to read more about this. There are few references, and thank you everyone for your patient listening.